Now, virus-free plants, already we have discussed about it. Now, plants can be affected by bacteria, fungal parasites and also viruses. Now, we have the chemicals to kill bacteria which are affecting the plants and the fungal parasites which are affecting the plants, but we don't have any chemicals to kill the viruses which are attacking the plant. For that, what we can do is we can develop by tissue culture technique, we can develop the plants which are free from virus. Virus will not be there, virus should not attack. Such type of plants can be developed. That is called the virus free plants. Usually plants are affected by fungi, bacteria, mycoplasma. Mycoplasma means these are the organisms which are intermediate of bacteria and fungi. They are not bacteria, they are not fungi, they are intermediate. Mycoplasma and the viruses. Now these are all commonly attacking the plant. Chemical methods, that is chemically we can control all microbes except the virus. That means what? Chemical method is useful to kill the fungi. By chemical method we can kill bacteria, we can kill mycoplasma, but we cannot kill the viruses. Now shoot meristem. Now already I drawn the picture. Now this is the stem. At the tip of the stem there is a meristem. Okay, a soft tissue is there, it is called a meristem, or a branch is present here. At the tip of the branch there is a soft tissue called a meristem, shoot meristem. Now the shoot meristem tip culture, that means the tip should be cut off and it should be cultured, that produces virus free plants. Now from that shoot meristem, if we are developing new plants, the new plants will not have any virus. Okay, virus plants, as viruses are not at all seen in the shoot tip. Now that means in the normal plant, the virus can attack any part of the plant. Root can be affected, stem can be affected, branches, leaves, everything. But the viruses are not at all attacking the tip of the shoot. That is a tip of the shoot, is always called a shoot meristem. So in the shoot meristem, the viruses are not at all attacking. So such a place we are selecting and we are culturing them, and finally we are developing new plants. So the new plants will not have any viruses. They are called virus free plants. Conservation of plant genetics resource. Now we have to see conservation, preservation, protecting. Now the plant genetics, genes of the plant, we have to call it as what? Genes of the plant. We have to conserve the genes of the plant. Now the genes are main resource because they are only producing the character. So we have to conserve the genes of the plants for future or for research. That is actually called the conservation of plant genetic resource. In that A, germ plasm conservation. Actually the plants or animals, anything if you take, the body is made up of two types of cells. I already told in the introduction. One is somatic cells, general body cells. Another one is reproductive cells like male gametes, female gametes. For example, in plants, the male gametes are pollen grains, the female gametes are ovules. So the pollen grains and ovules are called germ plasm of the plants. So the germ plasm, that means pollen and ovules must be conserved, must be protected, must be preserved. That is called a germ plasm conservation. Okay, for that what we are doing? Conservation of, that is protection of pollen, seeds, tissues, okay, for plant breeding. Now these are all must be conserved. We have to protect them for plant breeding, for plant reproduction in live condition. Then maintaining the viability. The viability means the way of growing. Okay, that is called a viable life activity we can call it as. Now how far the plant is growing well, how far the plant is healthy, everything we can call it as viability, viable. And fertility. Fertility means reproduction wise, the reproductive structures are very stronger. So the reproduction capacity is very high. That is called a fertility for hybridization technique in future. So in future we are going to do a hybridization technique. By this technique we are producing new varieties of plants. That is called a hybridization technique in future. For that the gene conservation is very important. Now the conservation, plant genetics conservation involved in gene bank. Now what is this gene bank? So now in the bioinformatics, that is in biotechnology, one part is our bioinformatics. all the scientists who are doing the research, they are giving the complete details of the genes of various organisms, starting from microorganism up to huge plants, animals and a human being. And that is a species wise. They are storing the information of the genes. Genes cannot be stored, but the information in the genes, what is that nitrogenous bases are there, how the nitrogenous base sequences are arranged, that complete details they are storing in computer. That is called a gene bank. DNA bank, that means genes are nothing but DNA. So the DNA bank can be stored, but in that original genes will not be there, but the information about the genes will be, gene or DNA will be kept of elite plant. Elite plant means high quality plants for biodiversity. Varieties of plants should exist on the earth. That is called biodiversity. For that, this is very, very important. Now, cryopreservation. Third is the technique by which whether it is a protoplast or cell or synthetic seeds or whatever it is, they can be preserved for a long period of time. It is a conservation, that is preservation of protoplast. I already told you what is the meaning of protoplast. Protoplast means a plant cell without cell wall. So the protoplast can be conserved, protected. Cells with cell wall. Cells can be tissues, group of cells can be preserved. Organelles, that means one cell. In the cytoplasm, we can find a number of organelles. What are the organelles? Mitochondria, ribosome, lysosome, nucleus. Many things are present. They are generally called cytoplasmic organelles. Shortly, we can call as organelles. Organs of the plant: leaf, bud, okay, node, okay, roots, primary root, secondary root, tertiary root. Everything you can call as organs of the plant. Extracellular structures. What is the meaning of extracellular? Everything is connected with the cells, organ, tissues, everything. Extracellular means there are certain chemicals not seen in the cell, outside the cell. They are called extracellular materials. Everything can be, that is preserved. That is conservation of protoplast, cells, tissues, organelles, organs and extracellular materials, we can say. The next one is extracellular matrix. Now, what is the meaning of matrix? Matrix means some jelly-like substance will be present. Enzymes, etc. can be stored. All the things can be stored at minus 196 degrees C. Now, first 0 degree itself is frozen temperature, but it is going for minus 196. Okay. Now, for example, some in the North Pole and all, we can find that the temperature is going down, where the ice will be present everywhere. Even there also, if you take minus 40, minus 45, minus 50 only will be there. But here the devices where the temperature can be maintained 196 using liquid nitrogen. By using liquid nitrogen, we can bring the temperature minus 196. In minus 196, we have to keep all these things, including matrix and enzymes, so they will be kept in good condition forever, for any longer period. That is the first one. Second one, no enzymatic action at such low temperature. Whenever the temperature is very, very low, inside the cell, inside the protoplast, inside the tissues and all, no chemical reaction. So what will happen? The protoplast, cells, tissues, organelles, organs and all will be kept in good condition without any spoilage. No enzymes will become active. Next point. Enzymes will become active when they brought to normal temperature. Now we are keeping all the things. For example, we are taking cells. The cells are stored in minus 196 degrees C. No action. The cells are kept in the same condition for long period of time, maybe 6 months or 1 year. After 1 year, we are taking the cells from minus 196 and we are gradually increasing the temperature. So the temperature is becoming normal temperature, our surrounding temperature, maybe 30 degrees C, 35 degrees C, 40 degrees C. Once the temperature becomes normal, enzyme action will be there. If the enzyme is acting on, the cell will become more active. If the cells are becoming active, we can follow the technique, tissue culture technique, and we can develop callus, we can develop new plants. So enzymes action will become active. Enzymes will become active when brought to normal temperature. Dimethyl sulfoxide, one chemical called dimethyl sulfoxide, glycerol, sucrose are used to protect them in minus 196. Okay, that all are kept in this minus degree, but they need some food. But the food materials, the chemicals must be kept. That they are called preservatives. So in the preservative, in this particular temperature, all these things will be kept in good condition, but inactive. That is called a cryopreservation. We are coming to intellectual property right. It is not connected with the technology. Now it is the rules and regulations. We have to take it as intellectual. For example, in our country, one scientist is doing some research in biotechnology, and the person, the scientist, is discovering a new thing. It is accepted by everybody. Then he is using his brain power. He is using his knowledge, and he is discovering. That is called intellectual property. That means he is investing his soft skill. He is investing his mind and knowledge for doing the discovery. So that intellectual property. Now it becomes the discovered, what he discovered, that becomes his property. That is his right to use that property. That is intellectual property right. For example, one scientist is discovering one new plant. That plant is the product of that particular scientist. He is having every right to give to anybody, either to the government or to the private party or anything. That is called a intellectual property right. That is IPR. Now we have to know something about it, IPR. Now once the person is discovering, okay, he is discovering a plant, then he has to get patent. Patent means license. Whether it is a state government or central government, he has to get license. He has to go and tell to the government that I have discovered this particular plant. So then the central government or state government will give license to the person. Okay, you are the authority for this discovery. But it's first we have to get the license. Once the license is obtained, then for the product, what product is discovered, he has to give one trademark. Okay, one trademark name should be given, and the trademark is meant only for that particular person. That is, trademark will be there, but that license will be given for a period of time. For say, for example, because the government only allowed the scientist to do research, so the government is giving license for 5 years or 10 years. It depends upon. So till that 5 years to 10 years, that person is the authority. He is giving one trademark name for that. Then after that, he is making it as copyright. That means others should not use the product. Without this person's permission, others should not use that. That is called a copyright. Then plant breeder's right. Suppose that patent, that person is having, the scientist is having license. He has given trademark. He is having the copyright. Nobody should use it. Suppose one business person is there, some other person. He is asking, I want to cultivate this particular plant. Can you give this? Now that person with a license can give right to this person. By taking some money, he can give. That is plant breeder's right. In the plant breeder, that means what, farmers or whomever it is, they have the right to cultivate the plant. Then next one, utility or model design. Now utility model design means how far the product can be manufactured and it should be taken to the trade. That we have to see. That is called utility model design. That is a trading, we can say. Geographical indications, whether the plant is growing in that particular geographical conditions very well or not, that the scientist must give. Suppose a person, a plant breeder, is coming and asking for this plant to cultivate, but the place where he is going to cultivate is not suitable. He should not give permission to that plant breeder. So even he has to consider the geographical indications. Then trade secret. Suppose the plant breeder or any other research people are coming and asking how you did the research, you give the secret of it. We will also try to do, means that is a trade secret, he will not give. If he is revealing that, that means everybody will give or will produce the same copy with that is what, different research centers. Then the discovery will become invalid. So he will not give the trade secrets. It will be kept in the form of secret itself. So the thesis paper, whatever he is doing, he will keep it everything in confidential. That is actually called the intellectual property right.